name is Jessica Nevarez-Mejia. I'm a graduate student at UCLA, and I'm this year's Immunology 2022 Travel Grant Grand Prize winner. And I'm here today with Dr. Joseph Beechem to ask him a few questions about spatial profiling and new technologies such as GeoMx and CosMx.
I'm Joe Beechem. I lead R&D for NanoString. I've been with NanoString about 12 years and been a key driver for us developing the spatial biology platforms, and it's fun for me to be talking with Jessica today about where spatial biology is going.
So it's, again, a pleasure for me to be here today, and I'm very grateful for this opportunity. So I do have a few questions for you today. A lot of them involve in GeoMx and CosMx. So the first question really is, GeoMx and CosMx technologies are now fully automated systems that can be used to analyze both protein and RNA expression, either at the regional level or at the single cell level. And so, what are some of the challenges that your team faced when developing these technologies, and how did you overcome these challenges?
That's a great question, Jessica. It's fun as we develop these technologies. You have to realize it sort of started before spatial biology sort of got all the buzz around it. It was like we were really looking at what are life scientists trying to do? What are translational scientists trying to do? Yeah. And I came from a background more on the next generation sequencing technology, so we were used to getting thousands of answers at a time. That's just sort of the way we developed technologies for that. Now, the trouble with that technology is that to make those measurements, you had to take the tissue out of the patient or to take the sample out of whatever system you're studying. You basically put it in a blender, you mixed it all up, and then you got thousands of answers out of it, which was great. And we really moved science farther ahead. Yeah. The trouble is it wasn't solving the unmet need of what we were really trying to accomplish in life sciences. One of the biggest things we're trying to accomplish in life sciences is how do we cure people from cancer? You take a problem like that, and it turned out that in the process of taking that cancer tissue sample and you grind it up, is that there's a whole lot of information that was lost in all the spatial context. So then, early on, and it was literally back in early in 2014, 2015, I was saying, how do we take the power of next generation sequencing, apply it to a spatial biology problem like a cancer tissue, and not have to grind it all up and blend it out, but we actually get all those high-plex answers directly in the tissue that we wanted to investigate. So that was the problem we were trying to solve. And you think long and hard on that, and you try to come up with an idea. How do you solve it in a different way? And what we did is everybody else was actually working on how do I put the assay in the tissue. Then the tissue goes into a microscope, and I try to deconvolve all these answers out of what I observed in the microscope. The fact of the matter, when I looked at that problem that way, is I'm never going to get 20,000 answers out of what I want to do by sticking 20,000 assays in there. Yeah. So then I had this idea, and I remember coming up with this idea. I was giving a talk at MD Anderson back in early 2016, and I scribbled this idea down on paper. And the idea was, instead of putting the assay into the tissue, all you really need is a sequencing barcode that basically rides on the back of all the reagents we're used to doing, no dyes on it or anything. And if we could somehow in situ measure that way, and then sort of the light that goes on is like, I put the sequencing barcode on those reagents with a photocleavable linker. Because light, what do microscopes do for a living? They shine light in little tiny spaces. I said, if we just did that, we could put 20,000 fish probes on at a time. And then when you want to know where they are, you shine light at a little tiny place, little sequencing barcodes come off, and we could, in theory, have unlimited plex on both regular fish probes or for antibodies. When you write down and you conceive an idea like that, to be honest with you, Jessica, usually I look at those ideas the next day and I'm thinking, oh gosh, that can never work. But when I looked at that the next day, I was thinking, I don't see why that can't work. Yeah. And, and then you come back in, and we weren't doing that. R&D, we all were trying to solve a different problem, bulk-based mid-plex digital quantization. I was able to talk one scientist into saying, okay, I'm going to try this crazy idea that night, say. And we had these ideas where you have a big idea and you try to act small to see whether it could work or not. Because if you've got a big idea that takes 5 years to figure out, it's not really going to happen. So anyway, this person had a big idea. We had this idea. It's a big idea.
Yeah.
And to figure out how if it worked or not, he literally took a tissue slice on a microscope slide, took little pieces of electrical masking tape, taped them up in a little grid on there, put the whole thing on a light box with a little pipette and just shine light, and then took the samples out by the little regions that were in between the electrical tape, put it in a pipette tip and said, can we measure the individual heterogeneity? And the fact is, the very first time, it did work with a pipette and a masking tape. And the signal and noise that we observed when we did it that way was about 25 times what we would ever do with no masking on a whole slide. And I remember when I first saw that data, I said, we're on to something then. So it starts with an idea, and you have to find somebody that you can think big, act small, see if it can work.
Yeah.
And it started working. It went from a 1-person program to 4 to 16. And then by the time you release a product line like that, you have about 200 people working on it at one time. Those are just the people here in Seattle in my labs. There's probably another 100 people outside that are working on it that we're paying to help us. And then you end up with a platform. It went from that idea on a memo pad, written down in a lobby of a hotel in Houston, Texas, in front of MD Anderson, to a released platform, fully released, commercialized in 3 years. So that gives you an idea.
Yeah.
That's fast. That's less than the time to do your PhD thesis.
Yes.
My guess is the idea went to a commercial platform, but that's sort of what companies do. We know how to, you got a good idea, we can make something happen in a hurry.
Yeah, definitely. And it's follow up to my next question is that you work with a lot of experts, like various fields for these technologies, and so you worked with the pathology, biology, chemistry, sequencing, and data analysis. So you have to put all these fields together to build this one product.
Yeah.
And how do you? So kind of you've already answered, but what are the beginning stages of bringing all these experts together, and what does this look like when you're developing this?
This is the whole reason why making a platform is so much fun. I mean, as a scientist, you can't ask for anything better because you actually have to, it's like sort of putting together like a fancy dinner. You need to have meat and potatoes and salad and dessert and wine. You have to bring all these parts together, and they have to work together to generate something really special. And that's what makes building platforms so much fun, because as a scientist, that means you got to have an understanding. All the people work together as a team, but the software people have to interact with the microfluidics people and the hardware people and the engineers and the optics people. And we have to, you have to be interacting with customers, because you don't want to build an instrument without customer interaction. So when we got a concept and we're starting to build it, we would literally bring customers in. And through things like granting programs like you are a part of, we call them technology access program, where people would actually say, here's the instrument we're trying to build to solve this unmet need in life sciences and translational sciences and discovery scientists. And then we would actually take real world problems from the outside world. And as we're building the instrument, real world problems would come in, and we would test them and debug real world problems as we're building the instrument itself. And that's been tremendously successful for us, so that when we actually build and construct this instrument to do something, it's ready to solve real world problems because we built the instrument solving real world problems.
And actually, that was one of the reasons why we decided to go and do the TAP program, because we saw very nice studies on cancer. And looking at the tissue and looking at this is a cancer cell or a cancer region versus a non-cancerous region, and you found so much data, and that is very fruitful but also very informative. Which brings me to my other question on clinical samples. So, spatial sequencing also allows you to get a lot of information from tissue embedded in FFPE. And this is important because a lot of clinical tissue or rare tissue cell types are often stored in FFPE. So what are your hopes for these technologies in the clinical setting, and how do you think that's going to impact traditional histological analysis?
Yeah, that's a great question, Jessica. I think we really did. I remember when we were first starting this, and when I was giving that talk down at MD Anderson, I was interacting with a good friend of mine down there. His name is Gordon Mills. He's an expert in sort of protein-based clinical biomarkers, cancer-based biomarkers, and understanding cancer. And he said, Beechem, whatever you're doing at this high place, it's got to work on FFP.
Yes.
Don't just make it work on some fresh frozen sample, because that's not really the samples of the world. The samples of the world, everybody that has a cancer in the entire planet, it's going to have an excisional biopsy. It's going to be put in FFP. And sort of, you can almost think of FFP as sort of the medical history of the world on a microscope slide. And when we developed this, from the very beginning, we made all the design choices we made on the chemistry, how we do the flow cells, how we do the instrumentation, was all built around that particular sample type, because that's the sample type that has a chance to change the world.
Yeah.
If and that was one of the key reasons why we did the FFP. The other thing beautiful about FFP is that even though it was very hard for, like, when I was growing up doing next-gen sequencing, it was always the hardest material, because you're putting the thing in candle wax, cutting it up, and putting it on a slide. It's not that great for nucleic acid extractions. But the way we designed the chemistry, we never take the nucleic acids or the proteins out of the slide or off the slide. Everything we do is in situ. And so when Mother Nature makes an RNA somewhere, I just leave it where it is, and I just image it where it is. And the same thing with all the proteins are there. And that design choice turned out to work out really well for us.
Yeah, and it's nice that you started with the hardest tissue embedding method, and so
because
everything
else becomes pretty easy.
Yeah.
Fresh frozen was easy once we had done that hard one. So.
All right. My next question is, as a leader in spatial biology, where do you see this technology in the future, and do you have an end goal for spatial sequencing?
Yeah, I think this is what's neat. When I look back over life sciences over the last 30 years that I've been in the field, there was a tremendous excitement. I think the biggest thing that happened in life sciences when next-gen sequencing came around, we really started to rewrite the book on how much we understand and how we understand cancer and a whole lot of aspects of population genetics and a whole lot of things. So it was a real revolution that took us all to a whole new place. We're right now at the early stages of the spatial biology revolution. Feels to me almost identical to what the early days of sequencing did. But then when I think of the applications where spatial biology can get to, I really feel like they're nearly unlimited. It's almost like you think of all life on this planet, we live in 3 dimensions, X, Y, and Z. Every plant, every animal that's on the planet, that is every bacteria, every virus, they all exist in a 3-dimensional space. And the fact of the matter is, we've never been able to allow them to be in 3-dimensional space and allow us to study it at this kind of high multiplex resolution and multi-omic resolution. So my feeling is that we basically have an opportunity to sort of rewrite the textbooks on how life on this planet works, because we're finally studying it without cutting it up and putting it into a test tube.
Yeah.
So I think I have super high hopes for this, for just having a better understanding how life on this planet works is one. That's sort of the most grandiose, if you will. But then the applications downstream of that are just sort of tremendous, because when you understand how tissues work, you can understand how they dysfunction in disease. And with this high-plex information we get, that means we can see dysfunction really early. We don't have to wait till you got a big tumor growing in your body to understand that something's wrong with your tissue, because we're measuring so many aspects of the biology of that. So I think, we're going to change the books, rewrite the textbooks in life sciences, which is great. But then I also think we're going to be able to invest the human condition in a really significant manner in that way.
Yeah, definitely. And the nice thing about genomics and CosMx is that you can look at a region or just like a specific niche in the tissue, but also look at the single cells within that region, which overall brings a lot of information, very important information for future studies. So what has been the most exciting part of your journey in developing these technologies and also bringing these technologies to the customers?
Yeah, no, I think that really gets to why do we all get into science and why do we do this thing for? I know for me, early on, it was just like a passion to try to understand how life is. I mean, how it's the most great mystery around. So you just want to understand, how does life really work? And that's sort of the beginning. And and that sort of took me all the way through a PhD at Johns Hopkins, and then I was a tenure professor at an academic place called Vanderbilt University. Mm-hmm. But what was weird is, as I was doing that, I have 3 daughters that were growing up, and I'm watching them grow up, and I'm thinking, I put all this energy into trying to figure out and develop technologies for life science, and I think I'm looking at my daughters, and I'm thinking, what am I really doing for them? Is there anything that's really significant? And that was sort of when I decided to leave academics. It was a great place. I loved being in academics. But then I wanted to make an instrument or something that would have impact, so that maybe if they're sick 20 years from now, I want them to be able to go to an instrument that maybe I helped create that's going to help them understand what may be wrong. And then, that's when I sort of went and started developing technologies and instrumentation, because you really want to help the world out in some manner. So then when you're asking what was the most fun for me at this stage of the game, is to actually give birth to a platform like that, and we were able to do that. And probably one of the neatest things was, I remember the very first 2 papers that came out of the GeoMx Digital Spatial Profiler through our Technology Access Program. Yeah, who's one of the co-authors on the very first paper is a person named Jim Allison, who won the Nobel Prize in medicine for sort of a breakthrough understanding of how to treat patients with immuno-oncology checkpoint inhibitors. And it was an absolute breakthrough, and he got the Nobel Prize, and I'm thinking to myself, wow, okay, we've developed a platform from scratch, and the Nobel Prize for probably one of the most significant discoveries in medicine is on our very first paper. Yeah. And then since that time, I think we have about 150 papers now, and they fill up the journal Nature, Clinical Cancer Research, and Nature Medicine, Science, Cell, and that's really the most rewarding part, because that's something that you're developing, something that's bigger than what you are. Now the whole world can make discoveries, figure out how to solve the next thing, just like your studies. You're trying to figure out how to actually help cardiac patients, and how to understand when they have complications and when they don't. To me, that's the most exciting part, is to see people like you doing the work that you're doing.
Yeah, hopefully we get that paper out soon also.
You will, I'm sure you will.
So how do you, in NanoString, how do you build a community within your R&D team or within the company?
That's a really good question. I mean, because when it comes to accomplishing things in science, I think it's all about teamwork. And in a company, it's even more important. When I was at academics, I had to bring in a certain amount of money on grants, and it was teamwork, but in a small team setting. Yeah. When you're in a company, everything is teamwork. It's like there's 280 people in R&D, but that's just what I do here. We're employing about 150 people in some other parts, and you got to get that whole community interactive and working. And then we also interact with people on the outside, customers like you, and all these other things. So how do you generate a large organization that can function and perform like a team? And we actually spend a lot of time on that. We actually have a process in here on the way we do R&D is something called an agile process. An agile process is you take your 280 people and you cut them up into specific teams, and the teams tend to be about 10 to 15 big. And they are set up with a clear set of autonomous goals that they are designed to solve. They don't have to ask me when they go to the bathroom. They have to figure out a problem, be able to act independently on that. Yes. And they have fun with it. Each of those teams give themselves special names. It's like crazy names, like Captain Marvel team, and then somebody else is doing something else. They all got distinct personalities, and then they all have autonomy and they can solve things, but they're all integrated and work together because that's the way this agile structure works. So, it's a real question and how to do those things. And in some respects, it's the difference between high-performing big teams that accomplish things and dysfunctional teams that really have trouble getting things accomplished. And I probably spend 60 or 70% of my time and efforts is making sure all these teams can become high-performing teams. So it's an important part of the whole equation.
Yeah. So that's actually a segue for my next question. So how do you evaluate success in your position, and how do you set effective strategies to meet your goal?
Yeah, no, it's really, it's a very important thing. Goal setting is really an important thing. And, we have a really good mechanism at NanoString. We have a set of what we call corporate goals, where it's like, these are the 12 things that we're doing at a company this year. And we make those decisions at the executive level. These are high-level goals that we put out. And then these things, what they call cascade down. So these things like, high-level goal, like one of the most important corporate goals in NanoString this year is the commercial release of the CosMx platform that we're in the middle of doing. So that is a high-level corporate goal. To accomplish that, you have to have all those individual teams be able to accomplish all these sub-goals that actually reach that one. Yeah. So we actually do planning that is what's called, you do the 1-year planning and you figure out that time horizon and all those things are worked out. And literally everybody in our company has set of goals written down, and those goals map to a corporate goal. So everybody knows what their goals are and how they contribute to the highest level aspects of the company. And it's not just an exercise, it's so much more rewarding to know this is what I'm doing and this is how it hits the big picture of what the company's trying to accomplish. So it's something that you sort of don't do in an academic setting, I don't think so much, but in a company setting, it's just so great. We got 800 people in the company. Every person in the company can tell you what they're doing and how that relates to the overall goals of the company. And that just makes it a kind of a fun place to work when you know what you're a part of.
It sounds fun to work in teams also.
Oh, absolutely.
To work as a team. So what advice would you give to graduate students such as myself that are seeking careers in R&D or in industry?
Yeah, I think, it all depends on, like for me, I spent a career in academics first and then went to industrial, commercial setting, if you will. And it really had to do with my own personal development, what I wanted to accomplish. I don't think it was really until I had my own kids that I was really thinking, what am I leaving the world? Up to then, it was all about, how I'm developing and how do I advance what my brain can do, say. And then it sort of transitions to how can I help the world out. But everybody develops on a different kind of scale. I don't know why it took me so long to figure out I really wanted to do something for the world, but it just took me a while to do. But I think somehow the academic versus the commercial sort of place you decide your career, I think is a secondary thing. I think the most important thing is you got to find, what is something that you can have the kind of passion for and say, this is what I wake up in the morning thinking about. And that's what you want to spend your life doing. What do you really have a passion for? So I think key thing in developing, find your passion, find that thing that you just drives you to want to wake up and do thing, because it takes a lot of energy to do science, right? So if you find your passion, that's the most important thing. And then you just have to follow that passion where it's going to take you. And it may take you initially through an academic career that moves over into a commercial environment, or it could go the other way. You can do things that are like hybrid things, where you actually do a postdoc at a drug development company. You could, so there's so many options now for a life scientist or for a scientist in general. I think back when I was doing my graduate school and things, I graduated from Hopkins with a PhD in 1986. It wasn't the same way. It was like everybody was just hammered through the academic. You're going to postdoc next. You're going to try to get a faculty position. And that was the way it was. But these days are very different. Fantastic science happens at almost all levels now. Doesn't matter if you're a company or an institution or not-for-profit. There's so many things. You just got to sort of find your passion, figure out what stage of development, what you want to accomplish with that passion, and just follow it.
Yeah.
And you'll change 2 or 3 or 4 or 5 times, but it's okay. It'll all be your own development.
Yeah, thank you. That's very helpful as a graduate student right now. So
Yeah, follow your passion is the key one.
and
Try to find nice people to work with.
Yes,
It's
yes.
not always easy, but in general, I mean, scientists are great people to work with
Yeah.
in general.
Yeah.
Yeah.
My last question is, aside from all of your responsibilities, do you have any hobbies or activities you like to do during your spare time?
Yeah, no, yeah, you got to have, I say you work hard, you got to play hard. So I got a couple of things I really like to play hard on. And what's weird is that some people don't think of what's an adjacent thing to a scientist, or you're a scientist. You know, when I think of the people I really like and I really relate to, I tend to really get attracted to people that are basically artists, creative people, artists, writers, things like that. So I feel like musicians, I find myself drawn to creative personalities, and I tend to find them in the arts. So that's where I got a passion for music. I have a passion for writing, and I got passion for reading, those things. So that's the art side. The other passion I really like is I'm addicted to Mother Nature, what she does in the outdoors. I'm one of those crazy bird freaks. I watch birds. My wife watches birds with me. I love to see animals in the wild. So you know, those are the two major things: being outside, and then just you know. I never stop reading books and listening to great music, and I love jazz, but I love almost all types of live music. But anyway, it fits, I think, with, people don't normally think of scientists liking crazy artists, but I think they fit together.
Yeah, I think so. Like as a scientist, you have to learn how to present your data. So you have to be artistic with that, right? You have to like know how to show your data for people to understand it easily. So I think artists is having being a good illustrator is another part of being a scientist. I think so.
No, and I think, you know, with your success of winning the grant, I mean, that was one of the key selection criteria. As you have a nice problem statement, you were able to communicate it really clearly on what you wanted to do, and the passion for you wanting to do it could shine through what your words said. And you're right. I think if there is a skill for a scientist, I think being able to communicate what you're trying to accomplish and be able to communicate to as wide an audience as you can is really an incredibly important skill as a scientist, because it's never going to show you wrong. It's always going to help you out. Oh, so great, Jessica. Do I get a chance to ask you some questions too, Jessica?
Yeah, of course.
Okay, so tell me, you're a year and a half or so away from getting your PhD done.
Yes.
And you know, tell me about how you selected your PhD studies. How did you decide to do what you're doing? And then what are you thinking about doing next, Jessica?
Yeah, so when I was an undergrad, I actually started as undeclared. And so I really didn't know what I wanted to do. I knew I liked biology since high school. I thought about going into nursing, but then I started the biology classes, you know, still undeclared. But I just really started liking how, you know, UC Irvine, where I did my undergrad, is a research university. And so when I was doing the introductory biology courses, I just really liked how my professors were doing research themselves. And I was like, I want to do that. I want to take advantage that I'm in a research university, and I want to do that. And so I was very lucky that Irvine has a minority access program, and so they have a lab there where you can join the lab and do training. It's a microbiology lab, but you get very basic training on like PCR, how to run a gel, and so I really enjoyed that. And I really liked research, and after that I declared biology my major, and so I kind of jumped around from lab to lab a little bit in undergrad. I did a little bit of microbiology, then I did a little bit of studying muscle, more molecular biology, and then towards the end I did cancer research in my last year. And then I really knew I wanted to go to grad school, and so I applied to UCLA, and I got into their molecular physiology program. And also in undergrad, I really liked immunology, and I never got the experience in undergrad to be in an immunology lab. And so when I started at grad school, I really knew I wanted to rotate in an immunology lab, and I just fell in love with it. I joined the lab of Dr. Elaine Reed, and we studied the sort of immune mechanisms that mediate chronic allograft rejection. And really, you know, what causes it is the antibody production is a strong humoral response, a lot of immune cell infiltration. It was just, kind of like very the core of immunology. And I just really liked it. And since then, I joined that lab, and I've been there in that lab. This is my 5th year, and I'm hoping to graduate next in a year and a half, and hopefully get in a career in industry. But I'm very open to doing a postdoc in academia, or a postdoc in industry. But I'm very open, and hopefully to stay somewhat in immunology and in transplantation.
Yeah, you can't ask for being a more exciting field than immunology, because it's shown to be the key of so many areas of health and disease, whether you're talking about your interest in transplantation, but it's also the heart of how we're curing cancer and developing immune active drugs to do that. And so many other aspects of the immune system are just intrinsic to sort of what makes us who we are. So I can't think of a better field to be in, to study. And it's very hot now. You're going to get lots of possibilities.
Yeah, no, and again, that's another reason why we did the Technology Access Program, because there's so many panels, immunology panels to choose from, that were super applicable to our studies. And so, again, we got so much information out of it, and we're still working on those studies and hoping to, again, publish them soon.
Hey, super exciting.
Yeah.
It's been fun to work together.
Yeah, no, thank you so much.